I'm going to introduce another gentleman who will be co-chairing this session with Dr. Stephen. And Harry, you'll be chairing the first bit of the sessions, and then Stephen will join you later on as we proceed. So I'm going to read out Harry's profile. Okay, as you've just seen in Dr. Stephen's presentation, Harry is one of the pioneer members of Synbio Africa. So Harry is an early career researcher and a trained biomedical scientist with degrees, a Bachelor of Science in Medical Laboratory Science and a Master of Philosophy in Molecular Medicine from the University of Health and Allied Sciences and Kwame Nkrumah University of Science and Technology respectively. He co-founded the Hive Biolab together with a team of researchers in the Open Bioeconomy Lab group at the University of Cambridge and Ghana to conduct research and create educational capacity for Ghanaian university students and researchers. Harry's current research focuses on developing a multiplex PCR diagnostic assay for detecting force-selected sexually transmitted infections in a single tube reaction using locally produced off-patent enzymes. His other experiences cut across open science and open source hardware for biology, entrepreneurship, and innovation. Harry, through his work, is growing and catalyzing the use of biotechnology and other emerging technologies like synthetic biology to scale social change in Ghana and beyond. Ladies and gentlemen, join me to welcome Harry Akligo to lead us to the biosensor session. Harry.
Okay, me again. So our first speaker is called Dr. Deepak Balaji from Synthetic Biology Center of South Africa. Dr. Balaji is a center manager of Synthetic Biology Center of South Africa. He's a chemical engineer by training and has worked in nanobiotechnology, industrial synthetic biology, and drug screening for precision medicine. The work resulted in patent. Deepak then went to EBM, Marie Curie Fellow, where he designed synthetic biovirus genome for recombinant protein expression. The work is the patent sold to the market as product of Geneva-based Switzerland. As a senior scientist at Oslo University Hospital, Norway, Deepak established the drug sensitivity screening platform for blood cancer. As center manager of CSIR Pretoria in biology, he co-founded he established drug sensitivity. He has 32 publications, a research paper, and he has 25 patents. Please welcome Deepak for the first stop.
Hello everybody, I am Deepak Balaji Thimiri Govindaraj, Center Manager for Synthetic Biology and Precision Medicine Center at CSIR. I am going to present on the CSIR technology platforms for synthetic biology and precision medicine, currently being established at CSIR. My work experience includes I studied in National University of Singapore and MIT Boston. I did an internship at Bioprocessing Technology Institute and Masters in program with the Merck internship program. I did my PhD at IMAC at VIB and I was awarded a Phelps Young Scientist Award for my PhD program. I did my EMBL Marie Curie Fellowship for 4 years at EMBL on a synthetic biology project, followed by a senior scientist program at Finland together with NCM, Oslo University Hospital, Norway, and Finnish Institute of Molecular Medicine, Finland. In this presentation, I will be showing about my technology platforms which I have established in the past and also my project proposal and current plan at CSIR Next Generation Health Cluster. During my PhD, I worked on a nanobiotechnology tool for cell surface omics, a Marie Curie for postdoctoral fellowship where I made synthetic baculovirus genome, and during my senior scientist program, I worked on drug sensitivity screening platform for T-cell cancer. Our orbital proteomics and for superparamagnetic based nanoparticles, we use a size-dependent nanoparticle interaction with the cells and the endocytotic pathway to isolate the subcellular compartments. For this, we manufactured nanoparticles by different surface coating. We tested their cellular uptake and used that for organelle isolation. How we did that is basically we synthesized 8 nanometer oleic acid coated superparamagnetic nanoparticles using thermal decomposition method, and then we performed ligand addition method to have a lipid coated superparamagnetic nanoparticle, and then also we did a ligand exchange method by having dimercaptosuccinic acid coated nanoparticle. As you see in this picture, initially these nanoparticles are coated with oleic acid and water, and they are dispersible in solvent. And when we did ligand addition or exchange, it became water dispersible. Using lipid-coated nanoparticles, we found that these nanoparticles predominantly sit at the cell surface, which are the green labeled ones, and with the membrane staining. And we use these nanoparticles to isolate plasma membranes with a simpler approach, where we culture the HeLa cells with the magnetic nanoparticles. We break open the cancer's HeLa cell lines and use magnetic field to isolate magnetic coated nanoparticles for mass spectrometric analysis. This work is published in Molecular Systems Biology in 2011, and we have a patent on this. We use dimercaptosuccinic acid using pulse chase methodology to allow these nanoparticles to get internalized over the period of 1 hour to 24 hours, and we observed that these nanoparticles could be used to isolate lysosomes at 24 hours. This has been confirmed through a Western blot analysis, where we observed that has strong enrichment of LAMP1 in the magnetic fraction, and also concentration of nanoparticles in lysosomal compartments in transmission electron microscopic images. During my postdoc work in EMBL, we made synthetic baculovirus genome using designer minimal baculovirus genome for gene therapy and genome editing using CRISPR technology. Why baculovirus expression system is that baculovirus expression system has been already used for recombinant protein production, like multibac, and also for OmniBac technologies. We also could make protein glycosylation by SweetBac or Glycobac. We used it for gene therapy, drug discovery, and virus-like particle production. However, the major limitation of baculovirus expression system is that most of these baculoviruses over the period of time age and results in defective virus, thereby decreasing the protein production capacity. Hence, we used approach called as SynBac, a minimal baculovirus genome minimization approach, where we genome annotated the whole baculovirus genome. All the green labeled ones are essential genomes. All the red labeled ones are non-essential genomes. Basically, the idea is to remove the red labeled genomes using homologous recombination. We broke down the genome into 6 parts, and then we used homologous recombination to remove the non-essential genes by iterative method. As you see in this original baculovirus genome, you have all these genes present in this fragment 1. After homologous recombination, you see a lot of genes were deleted. One example with the protein expression, we found the P10 was absent in the gel. And then by M-cherry expression, we found that the synthetic baculovirus genome, SynBac1, is more effective and with long-term stability and high protein production. And further, this technology has been patented. And been sold commercially by Geneva Biotech. Then I went to Norway to establish a drug-sensitive screening platform, precision medicine cancer therapy is a proof of concept. Why cancer precision medicine is that we can get tumor biopsies, people can do genomics, RNA-seq or proteomics, and identify drug candidates which could be tested in xenografts or patient-derived organoids, and identify drug candidates for cancer treatment. What we do is we establish a high-throughput drug screening platform which could be used to narrow down drug candidates. If it is an already approved drug, we can use it directly for cancer patient setting. I established this drug sensitivity screening platform for chronic lymphatic leukemia and multiple myeloma. Basically, we mimic the tumor microenvironment We cultured the cells in 3 to 4 viral format, and then we tested with 1 drugs, 10 drugs, and 20 drugs, and eventually scaled up to 516 drugs, and thereby we generating a heat map by comparing the different drug candidates' expression viability levels. What we have done is, as you see here, we have a platform already published in Leukemia and also in Blood Journal, where we cultured the cancer cells with the drug concentrations, 5 concentrations, and cultured them for 3 days for 72 hours and did cell viability and cell toxicity studies to generate heat maps and drug sensitivities. We further, using our monotherapy data, we did drug combination experiments and identified drug candidates which could be used for to identify synergy and also used for drug combination at lower concentration method. Similar approach we did for drug sensitivity screening in multiple myeloma for precision cancer therapy. What we did is we isolated bone marrow samples and cultured them with CD4 CD T cells and CD3, CD28, and use that to purify magnetically positive CD138 cells and use that for drug sensitivity screening with 30 drugs. And we also able to generate different heat maps by showing the drug sensitivity score. One of the critical things we identified is that when we compare the IC50 between all multiple myeloma patient donors between melphalan and melflufen, the red labeled melflufen is more effective than melphalan with respect to the IC50, thereby confirming our platform could differentiate the effectiveness of different drugs between patient samples. Thus, we did establish a drug sensitivity screening platform for relapsed and drug resistant patient samples where there is no therapy available. We did direct drug screening and we performed in vitro bioassays by serial cell proliferation and direct medium throughput assay, and we identified drug candidates which is currently validated by flow-based validation. And we are doing xenografts and from clinical trial. Our plan at the Synthetic Biology Center, Precision Medicine Center at CSIR, is to establish a directed evolution approach for superior tumor cell expression system, which is currently funded by ICGEB, and also to establish the lactobacillus-based synthetic bacterial genome for probiotics and recombinant protein production. And we are also establishing a cancer precision medicine platform for drug repurposing based on our experience in Norway to use high throughput drug screening on ovarian cancer and leukemia patient samples together with work package to microfluidics overcome check and investigate the tumor microenvironment and use that to identify potential drug candidates which we could directly test them in precision cancer therapy. And thus we confirm that we will use this platform to contribute to South African patients. Thank you.